So a lot of the fitness will be biased towards immune evasion, right? So again, fitness is defined by different things. But even for, I don't know if you saw that paper by Katrina Litko, where she was comparing, I think on one hand she was comparing DNA and RNA viruses that are transmitted like longer, like HIV and I think hepatitis. And mostly, I think it was realized that, okay, that's a different conversation. All right, introduction to phylogenetics. So we've talked about mutations. Some of them are bad, some of them are neutral, some of them are useful. Now we want to go into the phylogenetics now. We understand mutations, we understand spread, and we understand why genomic epidemiology is important. What is phylogenetics? So phylogenetics is the process of estimating evolutionary relationships, okay, among organisms by analyzing the genetic sequences. And the key principle here is organisms that are similar are going to be closely related. And those that are divergent, so we use the word divergent, which means different. So those that are different will not cluster together. Okay? That is the key principle of phylogenetics. The other important thing to think about here is the genetic or genomic distance, yeah, which is the number of differences between sequences.
Bien comprendre par la phylogénétique, c'est le processus d'estimation des relations évolutives entre les séquences du point de vue des séquences. Ici, c'est quoi? C'est que les séquences qui sont très proches, je vais dire, les séquences qui sont similaires, on va dire qu'elles sont proches, et celles qui sont divergentes, on va dire qu'elles ne sont pas proches. Et pour estimer la divergence, la similarité ou la divergence, on va se baser sur ce qu'on appelle la distance génétique. Le nombre de séquences qu'on a pour le nombre de séquences différentes de souches.
So, just to remind you, I understand these are difficult topics, not easy to teach. So if you have any question at any point, please stop me. Okay? Because what is important to me is that you understand the foundation. Rather than we rush to the end, we generate a tree, but you don't understand what all that means. Okay? Genomic distance? Don't worry, yes, we look at it. Okay? So the key principle is similar sequences cluster together, and they have something that we call more recent common ancestor. And sequences that are different will be clustering differently. In here, we have people from we have Kenya, South Africa, we have Burundi, we have, So if we were to take sequences from each of you, we'll find that the Burundians will cluster together first, right? But if you find Nehemi's sequence clustering together with Salva, then maybe one of them is lying. Maybe their parents actually came from a different country, right? Maybe. So anyway, that is the key principle. Sequences that are similar will cluster together, those that are different will cluster. And we use genomic distance as a way to estimate that similarity or evolutionary history. Okay? Good. So, genomic epidemiology, which we talked about, remember, we are looking at pathogen genomic sequences to track occurrence in a given population of disease, right? So we are using genomic population, genomic epidemiology, and these phylogenetic principles to track transmission patterns, right? Again, reminding you that not everyone in the population is sampled. We only sample some of them, okay? So looking at the transmission relationships, the genomic distance is used as a proxy, or we use genomic distance to determine how pathogens spread from one host to the other. So the genomic distance is like, if I compare two sequences, right? We expect that if we have 3 individuals, this one and this one, only 3 nucleotides are different between them. This one and this one, 5 nucleotide difference. So we can say this one and this one is closer to each other than this one and this one is, right? So we're using the genomic distances to understand the relationship between individuals, and also with that we get to understand the transmission relationships. Okay? Yes?
So I would like to know, all the distances, are they significant? If there is a difference of 1 nucleotide or 2 nucleotides, are all they are significant?
So you're asking the question like, how do you determine what number of differences are significant? I would say it depends. Because, again, let's say you could even have, so let's say you have 5 sequences. And often what we start with is what we call pairwise distance calculation. You could have that the difference between this one and this one is 1 mutation. And the difference between this one and this one is also 1 mutation. But those mutations occur in different parts of the genome, right? But because phylogenetic tree is context dependent, and you're looking at evolutionary history, it will have to determine that between these 2 and these 2, which one are they closest to? Like, between this one and this one, which one is closest to this one? And yeah, because then again, the position matters, and also relative to the other remaining samples as well. Okay? The other way I would also answer you is, it depends on the pathogen that you're looking at and which region of the genome. We do know, like DNA viruses, for example, Mpox. Because the evolutionary rate is quite slow, as Peter showed. Like we expect 2 mutations in a whole year, vis-a-vis 2 mutations in a month for SARS-CoV-2. So you can see, knowing what is significant depends on that mutation rate, right? Good?
Yeah.
Yes?
I think something else that has also been shown is for SARS-CoV, you have to look at the host. Is the host immunocompromised, or are you looking at the general population?
That's a very good point. Because the population, let's say even for monkeypox, it's okay. It's a DNA virus. The evolution is slow, right? But if you're looking at transmission within HIV population, that number can go up, right? Because the immunity is so low that it cannot suppress new mutations. So those mutations, probably most of them are neutral, but you see a lot more mutation than you would expect in an immune competent population, right? That's a very good point.
James, have you seen any interesting things in the children, the youngest patients,
For Mpox,
in terms of immune competency?
for children, some of it will also depend on like whether that's a vaccine preventable disease.
Well, I mean specifically for monkeypox. Like the youngest patients have a higher fatality, but do you see more mutations in the youngest patients?
But that depends whether it's grade 1A or 1B. I forget which one has higher case fatality. But then the issue also was, what they've shown is a lot of these cases, you see the baseline is often they are collecting only samples that show up at the hospital, right? So whatever denominator you're working with is biased because it's not like a whole population kind of denominator where you're getting samples both from the hospital and you're diagnosing random people in the population. So that case fatality rate is still being doubtful. And because they're also thinking, if I'm not wrong, like different sort of like estimates in like, let's say a place like Kinshasa versus, South Kivu, things like that.
It would be interesting to see if the likelihood of mutation in pregnant women or very, very young patients would be higher or more likely in women who are pregnant or very young patients.
I would expect there might be differences, that's for sure. Good point. Okay, so closer genomic distance. So when it's small, again here we are not saying small is 10 or 100. Again, remember, it depends. So we said it depends on the pathogen, it depends on the population. The third thing that I forgot to mention is time. Right? So we know the clade 1B outbreak started in September last year. So if we look at samples between September and December, obviously we are going to be talking a different set number of mutations if we look at now September up to now, October of 2024. That's a much longer time, right? So time is also important. But looking at the genomic distance, the smaller it is, it indicates more recent transmission, or you're looking at a shared outbreak source if you're typically thinking about bacterial context. But if you look at divergent genomic distance, which means like the distance is pretty large, so it suggests different transmission chains, okay, and also independent introductions. Okay, so similar to like, are you suspecting that this is a mutation that could have arisen from like a jump from another reservoir to the human, vis-a-vis comparing mutations between human to human transmission? Okay. So what is a phylogenetic tree? So this is a tree drew by Darwin in 1837, many, many years ago. So this is what we use to show the evolutionary relationships between organisms, right? So for a phylogenetic tree, we'll have different components. So we always say scientists are always confused because they want to name things differently, which is fine. So leaves, or taxa, or tips mean the same thing. Okay? So they simply mean, I would say they simply mean the samples we've collected. Okay? So these are the leaves, taxa, tips. Same thing. Sorry, I used to be a teacher in a deaf school. So sometimes if you see me doing things, I'm signing in my head. Sorry. So these are what we call the leaves, taxa, or tips. A, B, C, D, F. Okay? Then we have nodes. Nodes. So these are, okay, this is not the best tree to show external and internal. Okay, no, but we can. So nodes can be external, which means they are closer to the tips, or internal, which means they are farther away from the tips. Okay? But nodes are not actual sequences. They are not actual samples. They are hypothetical. They are ancestors. Okay? You understand?
Yeah.
So it's a sample which you've collected. And this one, did you collect this one? No. We compare the sequences and then we say maybe this parent to these ones had this sequence. So we are only assuming, we are estimating, not assuming, assuming is not the right one. We are estimating that this parent had this sequence. Okay? Then we have the branches, just like a tree, which correlates with the number of differences. Okay? We look at a time tree where the branch is slightly different. It's not just genetic distance, but also time. Okay? So in those trees, those are what we call time-calibrated trees. Okay? And then, most important here, we have something called the roots. So this is the parent of everything, the most recent common ancestor of everything, the roots. Okay? So for a rectangular tree, actually, mostly we'll talk about different tree topologies. You can read this tree going that way, ancestor to descendants. Okay? Any questions on this? So just important, that you know the different parts of a tree, the tips, the nodes, the roots, and the branches. Okay? Same as a tree. Roots, branches, tips, the leaves, the nodes.
In the trunk.
The trunk actually are the branches. Well, though I know in certain contexts, what they call trunk is the main branch. Yeah? But we don't want that complication at this point. So let's just stick to the basics. Okay? Les arbres phylogenétiques. Yes. All right. Now, phylogenetic trees are not transmission trees. We are looking at the evolutionary relationship. We are looking at transmission events. They are related, but they could also not be the same thing. Okay? They are related, but they could not also be the same thing. So just to be aware. Because for a transmission tree, you're looking at transmission events. And for phylogenetic tree, you're looking at evolutionary history. In well-sampled populations, actually they do mirror each other, right? But the fact that you will not collect all the samples, you cannot. The whole idea is you cannot make strong transmission conclusions on a phylogenetic tree. The fact that we have this sequence here that they share this node, right? And then we have these two that share this node. You cannot automatically say that this person transmitted to these two. Okay? That's what we are trying to say by distinguishing the two. That when you see a tree, you can say it's highly likely based on what you see. But then again, you have to consider different things, right? Are these two coming from the same location, for example? Because, this could be from Germany, this could be from Australia, and this could be from Kenya. This person has never traveled out of their country, this person has never traveled out of their country. So can you say this person transmitted to this one? Right? So just to make it clear that, those are two different things. Okay? Because it's one common mistake most people do. Like whenever they see any form of clustering, they say, this person transmitted to this, How many remember there's a very good study of HIV in Libya, where there were a couple of missionaries who came from, was it Netherlands? I don't remember. So a couple of missionaries went to Libya, and then there were a few, I think, were they kids that had HIV, and then probably one of them was positive, and they were accused that they had infected these people with HIV. It's a well-known case. I'll try to find it if I can. And those people were actually jailed for several years in Libya. And when they actually collected samples and did some phylogenetics, and looking at, for example, the time that these people came into the country, the evolutionary rate of the pathogen, it was just impossible that those people were actually the people who infected that child. There's also one known one in the US of a dentist and the partner, where I think the dentist was accused to have infected the partner, but when they plotted a tree and also looking at sequences across the whole community, it was just unlikely, because there were also sort of intermediate sequences in between. So anyway, again, they might be related, but they are not the same thing. That's to be aware. I'll try to get those 2 case studies and then we can look at them. But what can phylogenetic tree tell us about transmission? Obviously, the pathogen changes through time, and we are tracking those mutations across time. But remember, we are only sampling a proportion of the transmission. Then we are making a tree, right? So we are trying to understand the transmission through the phylogenetic tree, because often you don't have the whole sample set to get a transmission tree. So how do we build phylogenetic trees? How? We use DNA, RNA, or protein sequences, right? So we can use their DNA-based phylogenetic trees, RNA, or even protein-based phylogenetic trees. Important. So one, we have our sequences. And what do? We do an alignment. Okay? We align the samples A, B, C, D, E. We align them. So here you can see we have some mutations. Some are similar in all the samples. Some are, similar in only specific samples, and some are unique to specific samples, right? So we do our alignments. Peter did talk about masking certain regions, right? So some parts of the genome can be difficult to align, okay? Maybe because they have too many mutations or they have particular repeats. So when you're doing an alignment, you mask them. Masking simply means that I'm going to block them. I don't want them to be used in my genetic distance calculation. I don't know if you want to translate that.
Alors, bon, ici, avant de construire la phylogénie, il faut d'abord aligner les séquences. Et quand ils sont alignés, des fois, il y a des régions où c'est difficile de les aligner parce qu'il y a trop de mutations. Alors, dans ce cas, tu peux essayer de les masquer pour ne pas les utiliser dans l'alignement. C'est parce qu'il y a beaucoup de difficultés et ça peut vous induire aux erreurs. Donc, pour éviter ça, vous les masquez, vous les éliminez dans l'analyse. Non, on masque en fait, c'est pour ne pas les utiliser dans la suite.
I'll give you an example. Let's say I want to compare people from different countries. So this is our sequence data, right? I would say, well, maybe I look at the shape of the head, right? That can differentiate people. The color of the eye, right? But do you think we can use something like the height? The height is quite variable, right? Because even if we say people from this country are often short, but they're still tall people in that country, right? So masking is like finding a region that is not going to be very useful or very informative, and then you mask it, you block it, you're not going to use it, okay? And the third bit is now you make your tree. So first, you align the sequences. 2, you mask if you need to. Okay? You don't always have to. It depends. And 3, you get your tree. Simple.
Donc il faut d'abord commencer par faire l'alignement, comme des choses. Il faut aligner les séquences. Et après, si besoin, vous pouvez masquer quelques régions. Ça, c'est pas besoin, c'est quand on en a besoin. Et après, alors, vous générez dans le phylogenètique. Mais il faut commencer par l'alignement. Donc, she's asking, how do you mask the region?
Good question. So, Peter showed, did Peter show a bed file or it was just a VCF file?
VCF.
Okay. So, typically we use a file called a bed file. Okay? A bed file is sort of similar to, no, but we did use a bed file for primer trimming, right? I think he did show a bed file. So, we used a bed file to do primer trimming in epitope. So typically we also give a masking tool a bed file. And we tell the tool that I want you to ignore these positions. Okay? So typically we just give it bed file. Right? Okay, so
Bon, il dit que, voilà, il dit que normalement on utilise le fichier BED. Il y a un fichier qui s'appelle BED. Et dans ce fichier-là, on dit à l'outil, voilà, cette région-là, ne l'utilisez pas. Mais il faut l'indiquer, il faut utiliser le fichier BED. Et vous avez utilisé ça avec epidemiology quand vous avez utilisé les annonces. Vous pouvez voir les annonces que vous avez sur Madame Sikansa.
yes, I think yes.
Maybe just to link it to what we did last week.
Yeah. Did Peter show that? I'm trying to remember whether he showed it. So bed file is the same type of file that we use to trim primers. So it's the same file, but we give the tool and tell it like, okay, this position with this base, if you see it, chopped off. Okay? And it's the same file format that we use for masking. Just telling the tool, these positions, these kinds of mutations, ignore them. Like, as you're calling variants, you know, but this region, you know, just leave it out. So it's the same file format, okay? Now, to build phylogenies, we do use multiple methods, okay? There are many, many methods. The best method will depend on the data and the purpose. What do you want to do, okay? And don't worry, we'll talk about this. Often in bioinformatics, there is a trade-off. I don't know how we can translate this between speed and accuracy. The tools that are very fast are always not very accurate. And the tools that are very slow are often very accurate. In human context, they always say that the people who talk the most are usually probably not the cleverest, right? But I know that's a bad example. But anyway, can you just translate this like speed versus accuracy?
Voilà, donc là, C'est juste pour expliquer un peu la différence entre la précision et la vitesse. Là, il y a des outils qui sont très rapides, mais la précision n'est pas du tout élevée. Il y a d'autres outils, c'est très lent, mais ils sont très précis. Donc c'est avec ça, c'est ça qui a voulu. Voilà, ça dépend des objectifs. Des fois, ça se fait avec un rapport souvent, qu'on appelle le base calling. Des fois, tu peux faire un mode rapide et un mode qui n'est pas rapide. Quand c'est rapide, souvent il y a des erreurs. Et pour éviter, c'est rapide. I'm trying to explain using the Mpox system, the base calling.
Yeah.
The
Ah,
first mode
fantastic. Yes.
and the
So
more accuracy.
there's the fast, high accuracy, and the super accuracy modes, right?
Yeah.
Fast, have it, you get your results quickly, but the accuracy is down, right? And the super accuracy mode, you'll have to wait for longer,
Yeah.
but you know, you get good quality data. I think that's a perfect example.
J'ai une question. J'aimerais savoir s'il n'y a pas de script ou de logiciel qui permet de marquer automatiquement de voir ça automatiquement, genre un script et puis après l'alignement, là, le but pour utiliser notre logiciel. J'aimerais savoir s'il n'y a pas de logiciels ou des scripts qui font ce travail-là de façon automatique.
Quel travail ça fait?
Après l'alignement, là, oui.
Oui, c'est un outil qui va le faire. He's asking what kind of tools can you use to mask the job you want to use?
So this will happen with lots of tools. Even the squirrel that we use actually has a step where it masks certain regions. So it depends. Different. Even the what assembler do we use in epidemiology? Is it IVA? I think it's IVA. Yeah. Yeah. So many tools will do this. Like even for assembly, you can mask certain. It depends on what you want to do. So a lot of tools allow you to mask certain regions.
Donc il y a beaucoup d'outils qui peuvent faire ça, notamment le square que vous avez utilisé, le IVA, mais il y a d'autres qui peuvent faire ça.
Even squirrel masks certain regions because they know mutations in this region are problematic, you know, things like that. Okay, something we talked about this morning, the results that you will get is very, very dependent on the quality of the data. Okay? So let's say you use low quality data, probably you'll get many mutations, right? And that will determine, you know, how your tree looks like. But if you get good quality data, you're also very sure about, you know, the results that you get. So these are just very important things to consider. So in general, we start with an alignment, and then we get to the tree. And I want to just point out, I think this is a very nice way of visualizing. So if you look at A, B, right? And the circles are mutations, right? So A and B share green mutation, yellow mutation, and blue. Do they have any mutations between them? Per difference, A and B. Hakuna differences? No differences between these two, right? I'm speaking 3 languages at a go. So you can see that here we don't have any branch length between A and B. Their most recent common ancestor is this, but they are exactly identical. So you can see that. When it comes to phylogenetic trees, remember the vertical lines are only used for visualization. They are not important. Please, can you insist on that? In French. Vertical lines, not important. Horizontal, very important.
Les lignes verticales, c'est juste pour la visualisation. C'est pas important.
Yeah, it doesn't matter. Like, if this was this short or this long, doesn't matter. Yeah? Okay. Now, when we look at C, we can see A, B, and C have that branch length and that branch length, right? And this is because if you look at A and B, they share this mutation, they share this mutation, but C has a mutation that is different from the two. So you can see that, C has this mutation that is different, okay? Now, if you look at the branches, but A, B, and C share a most recent common ancestor here, right? So and because they share this, right? We are going to ignore this for now because it's present in all of them. We can see that C is different from these two by the green one. So these ones share this most recent common ancestor. This mutation is on this branch. I want to go slowly so that we understand this, okay? So this mutation, the green one, is unique to these two sequences, okay? Because it's only present in these two. This mutation is unique to C because it's only present in C, right? But here, orange is present in A, B, and C. So it's on this branch. So in phylogenetics, when we look at time, we say that this sequence acquired this mutation, okay? It acquired the yellow, don't know whether it's yellow or orange. I'll use orange. And then C acquired this red mutation, while A and B both acquired the green mutation. Okay? Now, D and E, they share this mutation. So you can see that mutation here. But E has an additional mutation, this pink or red. But you can see that this red is at a different position, it's different from that one, right? And you see, unlike A and B that had no differences, D and E do have a difference, right? So it has this red mutation here that makes D and E different. But they do have a common, this, assume that this color is the same as this color. Sorry, my probably was color blindness making this one. But this color is the same as that. Okay? So they do share this mutation. Then when you go back in time, all of the sequences have this mutation. Right? So when we look at these branches, what we are representing are the mutations that are occurring along those branches. Here, we are using just like a single mutation. But you see, often when you're looking at a whole genome, we are talking about many mutations, so it's not just one. Okay? Any question? Please ask any questions here. Because this is important to understand before we go any further.
Donc, violet, si on prend la dernière colonne, ici c'est ancestor. But before on appelle le violet. Violet c'est quoi? Et c'est rouge. Donc le violet, entre le violet et le vert,
Ça?
oui,
Mhm.
et le violet,
Ça?
yes.
Mhm.
Quel est le plus vieux ancestor?
Yeah, so this is present in all of them. So it will be present in the root. Okay? So that's how, like, and remember, when you build a tree, typically you're not starting from this to this. You're often like working back. Look, remember, you're establishing the evolutionary relationship. So how this often works is that we have all the people in this room, right? I start with, let's say, Kisti. I compare. How many mutations? Okay. How many mutations? Okay. How many mutations? Fini, right? Then I go to Davis. I repeat. How many mutations? How many mutations? Then I go to Tony. How many mutations? Then from all that, and now I start looking at, okay, how are people related to each other? Okay? Then now I start working backwards until I go to the roots. Okay? We don't start by, saying, oh, which one is common to everyone? And then we start from the root going that way. So think of building a tree, you're working backwards in time. Okay? Which example? So, I mean, if you think about, let's say, if I collect human samples from everywhere in the world, right? Then I will do genetic distance comparison. I will expect that people from Burundi will be closer to each other, right? People from DRC, Republic of Congo, Kenya will be. And then, as we work backwards, maybe we'll see that people from West Africa are closer to each other, right? So, and we expect Republic of Congo and DRC will be closer to each other than Republic of Congo to, for example, Tanzania, right? So we are going back in time, right? And then we get to a level whereby we expect that people from Africa will make one cluster and people from Europe will make another cluster. Okay? So remember, we talked about scales, different scales. So let me. Now, if we look at the map of DRC, right, we have different provinces. There's Equator, there's North Kivu, there's South Kivu, right? Right now, when we collect samples, we expect that South Kivu samples will be closer to each other than South Kivu and Equator. Like, if you collect 2 samples from Equator and 2 samples from South Kivu, we expect that, based on geographic distance, that the 2 samples from South Kivu will be having smaller genetic distance than South Kivu to Equator, right? So it's the same concept that, we expect that as you build your phylogenetic tree, so you're starting from more similar to bigger distances as you work back through time, as you go from the internal nodes, from the external nodes to the root. Okay? So the way I would look at it is this. If we go back to our phylogenetic tree here, A, B, genetics distance, 0, right? Because they are similar. A, C, how many differences? Yes, between A and C.
2.
says 2. Any other? What is the genetic distance, which is the number of mutations between A and C?
Alors, bon, là-bas sur les images, chaque couleur représente une mutation. OK? Il a demandé entre A et B combien de différences de mutations là-bas.
0.
Combien entre A et B? Il n'y a aucune différence. Donc c'est ça la distance. Il y a 0, c'est 0. Maintenant, entre A et C. A et C, combien de différences?
A et C. C'est
A et C.
quelle couleur?
Attends.
Vert? Eh? Oui. Et rouge. Oui, because this is one mutation or set of mutations. In this case, we only have one. And here we have another difference, which is 2. Okay? So these have only 2 differences between them. Okay? Good. Now, if we were to look at what is the distance between D and E?
D et E, combien de différences?
1, right? Okay, fantastic. Now, what is the difference between A and E?
A et E.
5. Okay, tell me which are the 5?
Which can come to show you there.
Which? Okay, come. Show.
We have
Yes, yes.
this one.
That's
This color. One.
Tu touches tout simplement.
Green. Green. And yellow 4.
You change your minds. Okay, she said 4. Is that correct? Cut? So, I mean, you could also just look at the branches. So there's 1, there's 2, there's 3, and then there's 4. Okay? So 4 mutations between them. So think of that as genetic distance.
Yes, sir. It is good for look the tree or the look the alignment.
It's better to look at the branches. The branches is easier.
This is easy.
Yes. But again, remember this represents this.
Yes, but what is better?
Good point. If you have a good quality alignment,
Yes.
just use the tree. Yes.
Yes.
So the tree will show you the number of differences. Again, I want to insist that remember that here we are only showing one mutation on each branch. But normally there'll be like many mutations. So not just one. Okay? The other complexity here, we are not showing shared mutations across different branches. Sometimes there are things we call convergent mutations, whereby you can see a mutation here that you also see here that just happens by chance, and it's not an evolutionary relationship. Okay? For example, for pathogens where you have multiple circulating clades, okay? sometimes what happened in the evolutionary pathway of that pathogen, they face what you would call similar evolutionary events. Then they both acquire that mutation independently. It doesn't mean that, the most recent common ancestor for all of them have that mutation. I know that is a bit complex, and we can talk about that. Like, let me give you an example. We have, good. We have clade 2B for monkeypox, right? And we have clade 1 for monkeypox. And before, we used to have jumps between the animal reservoir into the human, right? And what has made monkeypox now have human-to-human transmission? ApoBec mutations, right? But ApoBec mutations, now we can say have been acquired independently by clade 1B and clade 1A. If we look at the root of monkeypox, does it have ApoBec mutations? Example. Let's assume this is clade 1B, this, right? Before monkeypox outbreaks, there had to be a jump from, like, let's say, a rat into human, rat, human, rat, human. But now we have human to human, human to human, human to human. Pourquoi? Parce que la mutation ApoBec. Have you heard of ApoBec mutations?
Il y a un type de mutation qu'on appelle ApoBec. Ce sont des mutations. I don't know about ApoBec.
Okay, let me explain. There is a reason why we can have the same pathogen in humans and in animals, but that pathogen does not survive pretty well in humans because it does not have the mutations that allow it to be efficiently transmitted between humans.
Bon, en fait, il faudra savoir ça après. On appelle ça ApoBec quelquefois. Mais bon, ce qui est d'expliquer, c'est quoi? Vous pouvez avoir un pathogène dans un animal et dans l'homme, n'est-ce pas? Mais la transmission de l'homme à l'homme, ça ne lui permet pas de bien parce qu'il n'a pas de mutation requise pour être transmis d'une manière efficace.
Yes, because remember, animal cells are different from human cells. So, for a pathogen to adapt to a human environment, it needs a different set of mutations, right? So you could have an instance where there is that jump from an animal to a human, but then it cannot efficiently transmit from that human to the next person. Okay? Now for monkeypox, there are these mutations that they call ApoBec. So once these pathogens had this ApoBec, then it's been easier for the human-to-human transmission. Okay? Now, if you look at monkeypox, we had the ApoBec mutations only in clade 2B. In clade 1, we didn't have ApoBec mutations. Then in the recent outbreak, in clade 1B, now these pathogens have also acquired ApoBec mutations. And now they are also seeing efficient human-to-human transmission, right? Good. When we look at a tree, we often say this is the most recent common ancestor. The mutations that are here, often you expect that they are in all these pathogens, right? And you typically expect that the set of mutations that are here are very different from the mutations that are here. Correct? But sometimes you see a mutation that is here and is also here, but is not here. Because we expect that the mutation that is here and here, we expect to see them here, because they should be shared by everybody. Okay? But in the case of Appleback, these have been acquired independently. It's not part of the evolutionary history of the pathogen. Okay? So that is what we often call convergent evolution. Let me just type it here. Convergent evolution. Okay? This one.
Yeah. Alors, il essaie d'expliquer en fait un autre concept qu'on appelle les mutations convergentes. L'exemple qu'il a donné, vous voyez, le bleu, c'est une mutation qui est partout. Le bleu, c'est bleu ou bien? Bon, c'est bleu ou violet, quelque chose comme ça. Donc, c'est une mutation qui est présente dans toutes les souches. C'est ça. Mais des fois, tu peux avoir une mutation, par exemple, au niveau de AB. C'est une mutation qui est également au niveau de E, D, mais qui n'est pas commun. Donc, ça ne fait pas partie de l'histoire évolutionnaire de l'agent pathogène. Et c'est ça qu'on appelle alors les mutations convergentes. Donc, cela, il faut être un peu attentif. Ça peut être une mutation qui est l'ancêtre commun, qui est partout, mais il y a des fois des mutations qui sont dans un petit cluster en haut, qui sont les mêmes, et en bas, mais qui ne fait pas partie de l'évolution historique de l'agent pathogène.
Yeah, so
C'est ça qu'on appelle alors convergent, mutation convergente.
you see a mutation here that you also see is present here, but it's not in the root. So that's what we call convergence. So these pathogens and these ones have acquired it independently.
Donc ce sont des mutations qui sont arrivées, mais d'une manière indépendante.
Dans cette commune, on va pas.
Donc, par exemple, toi et moi, on peut avoir la même mutation, alors que notre ancêtre commun n'a pas cette mutation. Mais toi tu l'as eu, au même moment je l'ai eu, mais notre père n'a pas cette mutation. Normalement, toi tu as une mutation, moi j'ai une mutation, notre père a la mutation. Voilà, mais deux fois, toi tu as une mutation, et moi aussi j'ai cette mutation, la même, sans que ça apporte aussi à notre père.
I think, is it, maybe if I explain this correctly, I might be forgiven. But if you think about a bat and a bird, right? A bat is a what? Is a mammal, right? And a bird is not a mammal, right? But both of them have wings, correct? Would you want to share, would you want to say that because birds and bats share wings, then they are much closer to each other than a bat and a human being? A bat and a human are mammals, right? And a bird is very different, right? But a bat has wings. A bird has wings. So, can we say that a bat is closer to a bird than it is to a human?
Il a dit, si je peux essayer d'expliquer ce qu'il a dit, il nous dit que les chauves-souris sont des mammifères, comme nous les humains. Mais les chauves-souris ont des ailes, sont capables de voler. Est-ce que pouvons-nous dire que du fait que les chauves-souris ont des ailes, ils sont plus proches des oiseaux, par exemple, que de nous. Aujourd'hui, je sais pas,
Non, non, mais il faut suivre la chose. Là, qu'est-ce que les avocats ont fait? Il donne un exemple, n'est-ce pas? Trois choses. Il a donné la chauve-souris. Il a donné l'oiseau. Il a donné l'homme. Maintenant, la chauve-souris, elle a des ailes. La chauve-souris, elle a des ailes. L'oiseau a des ailes. L'homme n'a pas des ailes. Et puis il a dit, la chauve-souris, c'est mammifère. L'homme est mammifère. L'homme n'est pas mammifère. Alors, est-ce que la chauve-souris, elle est proche de l'homme ou elle est proche de l'oiseau? So they're saying that is close to the pet.
Maybe a different example. So a bat uses sort of echo hearing decay, right? So it projects sound and the sound wave come back, so it knows where objects are. A whale in the ocean also uses that same process. It uses that echo sound to sort of see different objects in the ocean. But a bat and the ocean are in two separate environments. One's in a cave, one's ocean. They have never come into contact with each other before, but yet they use the same echolocation. So that is evolution. They reach that point independently. Does that make more sense? So what he is saying is similar in that virus. Like when you have clade 1A and clade 1B, they're in different environment, different culture, and they both receive that same mutation, but they were sort of done at separate stages or independent of each other.
I have a good example now. What I was trying to say with regards to Appleback. Clade 1B acquired Appleback. Good human-to-human transmission. Clade 2B says. Now we have clade 1B that also has Appleback. Okay? Now, for example, can we say that this clade 1B here that has Appleback is closer to clade 1B than clade 2B is to clade 2A? No? Right. We can't say that. We can only say that these two clades have acquired that mutation independently. Like that Appleback is not in the root of these two, right? But as Tony was saying, based on the current prevailing environment, they acquire that mutation, and the pathogen, accidentally, is finding out that having that mutation enables efficient human-to-human transmission. So the pathogens that have those Appleback mutations are becoming more successful, right? So this is convergent evolution, very independent, okay? It's not present in the root. Now you've understood? Yay, good stuff. Okay, I think we better stop here, go have lunch, and then we can continue.